Dr. Ivan has made all the way from Washington, D.C. Here. So I would like to briefly talk about scientific path of Ivan. Ivan's scientific path. Ivan received his PhD from Novosibirsk State University, and then he moved and did his postdoc at the University of Berkeley. And then he moved to the Lawrence Livermore National Lab. And actually, while his PhD was in physics and mathematics, he switched the field of study to genomics. Specifically, I think his research has been dedicated for a number of years, maybe for the last 20 years, to study regulatory elements in human genome. Ivan used to work on so-called gene deserts. And right now he's working on gene regulation, on coding DNA regions, enhancers, but recently he actually started to work on silencers in addition to enhancers. And his lab right now at NIH, where he moved in 2007, his lab is really a very dynamic lab. I know his lab while I was there. The lab has developed a number of different methods and web servers which are available. And today he is going to tell us about, I think, one of the most recent projects on silencer variants. Thank you, Ivan.
Thank you so much, Anna. And thank you so much for the introduction. And thank you very much for the invitation. I guess you're all used to Kingston by now, but for people who came here for the first time, it's truly a magical place. You have no idea how beautiful it is for somebody who just turns right from the highway and enters this beautiful city of Kingston. With that, let me go back to science, and as Anna said, my lab works on silencers. We work on non-coding DNA, and I'll try to maybe convince you today that our genome is full of non-coding DNA, but not all of it is junk. The genome has been sequenced many years ago, right? In 2001, we've got the full sequence of the genome, 3 billion nucleotides, 30,000 genes. Everybody is excited and happy. However, only 2% of the genome is coding. And 98%, as we know by now, is non-coding. So what does it do? Obviously, it's junk, right? All the pseudogenes, microsatellites, replicating DNA, that's all in there. And maybe we should just ignore it, focus on the 2% that makes sense, and live a happy life.
Can you just click on OK?
I'll try to. I think I'm gonna go off the full screen for a sec. Try to click here.
And OK.
And okay, one more okay. Okay, sorry about that. So my good friend Marcelo Nambreger, who is now in the University of Chicago, he said, okay, why don't we prove that the junk DNA is truly junk? He decided to delete the longest intergenic intervals, this non-coding DNA between genes from the mouse genome. He succeeded in doing so just 3 years of his postdoctoral career. And he's got these 2 beautiful mice lacking the longest gene deserts in the mouse genome, 2 megabases, 1 megabase bacterial genomes out. And what happens? Absolutely nothing. These are viable, healthy, happy mice, exactly the same as the wild type, hundreds of biochemical tests, no difference whatsoever. Proven so nicely that non-coding DNA is purely junk. However, if we look at it from a different viewpoint, I'm sure a lot of you have heard know about lactose intolerance or lactose tolerance, depending on how you look into that. And 2/3 of the human population are lactose intolerant, or 1/3 is lactose tolerant if you look at that. And this phenotype is associated with domestication of cows that took place primarily in Europe and Northern Africa, where now the population is mainly lactose tolerant. And this is a tiny switch. In the European population is a C to T change, and in African population there are 4 changes located in the non-coding DNA, not in the lactase, but a nearby gene, suggesting that these tiny changes could have quite a profound phenotypic impact on what's going on. As time passed, what we've observed is that this is not the only non-coding change that has a phenotypic effect. There are tons and tons of changes, and this is a famous sonic hedgehog enhancer located a megabase away from the gene and so-called ZRS enhancer that's associated with thumb polysynderectomy. And that's, again, that's not the only disease with a severe phenotype that we can link to these non-coding mutations. Obesity, Parkinson's, Hirschsprung, type 2 diabetes, the list of diseases linked to these non-coding mutations keeps growing and growing. If you look at GWAS overall, what you will observe is that only 5% of GWAS variants are coding in nature, and the vast majority are non-coding. They reside in what we might assume is junk DNA. One of those GWAS variants is located close to alpha-synuclein. This is the gene that's associated with the risk of Parkinson's. And in the wild type, this distal enhancer is suppressed. However, an A to G mutation prevents efficient binding of a repressor AMX2 and KX6.1 complex to this distal enhancer. The enhancer reactivates, the alpha-synuclein level of gene expression goes up, and that increases the risk of Parkinson's development. So how can we distinguish between truly junk and non-junk within our genomes? And my lab is a computational lab. We are not allowed to touch a pipette. And we are developing deep learning methods in order to identify these functional variants in the human genome. We use deep learning. We rely on multiple layers of experimental characterization of the human genome. And we collaborate with labs that perform experimental testing and can either validate our computational predictions or completely reject those. And in the recent years, we have been able to link non-coding mutations to a list of diseases, including autism, type 2 diabetes, B-cell lymphoma, and they'll touch base on the last two today. The model, it's a deep learning model. I'm not going to go into the details, but an interesting part of deep learning models is that they are these crazy millions of parameter models that somehow train themselves. You drop everything that you have into a black box. And a very accurate model theoretically is going to emerge that knows absolutely everything about everything. That's how they seem to be working. In our case, it was a model of 143 million parameters that we trained on the ENCODE data, and we've been able to establish a so-called second language of the genome. So when you have coding DNA, that's the primary first language, and non-coding DNA supposedly has an encryption that we detected, and many other groups as well. We are not unique in this aspect. And we've got k-mers, we've got motif combinations, we've got something that we believe is a grammar of the non-coding part of the genome. And with that, we have been able to build models of tissue-specific enhancers. Again, I'm not going to go necessarily into the details, but it works when we grab ENCODE and roadmap epigenetics data, when we get enhancers from multiple tissues. We get models, we can characterize how accurate those models are. And when we talk to computational people, what we love to do, we love to look at the area under the curve, UPRC, all these metrics. And the way you look at it, the closer it is to the number one, the better it is. There is a cross-validation, there is proper hiding of your training data. And when we look at how well we predict the regions of open chromatin, of keystone modification, of binding of transcription factors, we do well. And how well is it? that well? How well do we predict enhancers? And what I'll tell you is that my lab has been working on this for dozens of years. And the best we could do is this. That's called SVM, so support vector machines. When we were accurately modeling everything, our accuracy was around 70%. As soon as we dropped all our data into this deep learning, it jumped immediately to 85%. And these days it's 95%. We have no idea how it works, honestly, but it works so well. And if it can predict regulatory elements that well, what we can do is to perform a so-called in silico mutagenesis of regulatory elements. We can take an enhancer, a regulatory element, and we go and mutate every single nucleotide 3 times, and we repeat it for all the nucleotides within this enhancer. And if this is the score that we compute using deep learning, then we can detect cases where the score changes a lot. And for every nucleotide, we can plot this change score. And the positive scores, those that are high, they correspond to negative impact on the regulatory element activity. I'm sure you've dealt a lot with transcription factors, and those are important, but computationally predicting binding sites of transcription factors has been challenging. And please trust me on that. It's really, really difficult to find where they are. It's much more difficult to find boundaries of transcription factor binding sites. With deep learning, as you see from this plot, it's very obvious where the boundaries of the binding sites are. So that was the first thing that appeared from our deep learning, that we detect binding of transcription factors well. And if you look at a regulatory element, if you look at the mutational profile, and if you start applying this method that quantifies how damaging every mutation is, you see peaks and you see valleys. And what do they mean? When we overlap this profile with non-binding of a transcription factor that binds to that particular enhancer, we immediately saw that 2 of the tallest peaks, these are non-binding sites of HNF4 alpha. It's a liver enhancer, so it's not surprising that HNF4 alpha binds there. However, when we looked at other places where we see peaks, we see binding of other transcription factors. It's not necessarily that we know what binds there. We can identify within these regulatory elements active sites and inactive sites. And that's fine. How does it correlate with experimental data? And what has been published a few years back are so-called reporter assay QTLs, where you look at your classical eQTL, and instead of looking at the level of gene expression, you try to measure the impact of mutation on the activity of regulatory element. And a few thousand, 20,000 QTLs have been reported, and these are variants within the human genome that do change activity of regulatory elements. Can we get them? Do we get them if we have these models that I'm telling you are very accurate? And if you look at the density of QTLs across the genome, that's where they are, in the areas of open chromatin. When you start looking at the enhancers are functional elements. You expect regulatory mutations to be within them. The density jumps. But when you look at those sites that we predict as transcription factor binding sites, you see a tenfold enrichment. So we accurately get those. However, if we slide through the top scoring positions within enhancers that we predict, this density of QTL enrichment goes really, really high, suggesting that if we predict something as a functional element, we predict something as a functional mutation, and it's top scoring, it is very likely to be top scoring in the experiments. And this is the collaborative work with our colleagues at the Berkeley lab. I told you there are peaks, and if we see a peak, it's something that damages the enhancer activity. We saw tons of valleys where we predict a mutation has a strengthening effect on a regulatory element. And that's kind of surprising. It's like, why would a mutation improve something? You are mutating a functional block within the human DNA. And yet there are places where we saw strengthening of activity. And we were leaning towards throwing them away until the Berkeley lab went into the experimentation. And that's the ZRS enhancer that I told you about that's linked to polysyndactyly. They went through that enhancer, and instead of mutating every nucleotide computationally, they decided to do that experimentally. And they've done this through enhancer mutagenesis. You see the staining in the limb, and you see staining profiles. And what they have been able to demonstrate is that 71% of enhancer mutations strengthen enhancers. And that was quite shocking at that time, and still quite is, meaning that in our enhancers, within them, they have propensity to become stronger and to do something else. And this, oops, sorry. And this is going to be important for what I'm going to be telling you about later on. I'm sorry. I have many that So with that, I'm going to shift gears towards the diseases. So who cares about these computational models? What we have been able to do is to grab these models and to start applying them to specific diseases. And we, in this case, we joined forces with the lab of Francis Collins from the Genome Institute, and we started looking at type 2 diabetes and patients with type 2 diabetes. Francis Collins lab, they had data on pancreatic islets. They had regulatory DNA that they characterized and extracted using ChIP-seq experiments. We grabbed that data and we built an enhancer model, and pancreatic islet enhancer model. And we started asking questions. If that's the model, can we pull out type 2 diabetes disease causal variants out of the GWAS data? And you will tell me that we don't even need to do so. GWAS studies produce these variants. We just need to look at them, and here you go. However, there is a huge difference between association and causality. That's why GWAS is called GWAS, not GWDS, or whatever it is. It's all associated variants. And the vast majority of them do absolutely nothing. So how do you look through all these variants and figure out those that cause something, that do something? And we believe that by applying our model, we should be able to detect disease causal variants. And we used some other experimental data. We used MPRA data to demonstrate that what we predict is having an impact on pancreatic islet enhancers is indeed having an impact from the experimental data. How does it look when we compare our model to MPRA or MPRA experimental analysis of enhancers? This is one of the enhancers, and this is the mutational profile that shows up from our computational work versus something that you get from the experimental enhancer mutagenesis. I would like to point your attention to these two spots. According to the experiments, these are two nucleotides that have the most drastic impact on the enhancer activity. If you look down, that's exactly the two spots that our model predicts. So in many cases, we can accurately predict disease causal, or rather enhancer damaging variants. And if we compare our model to other models, we do well, and usually we do better than other models. Let's go back to GWAS. So this is type 2 diabetes. It's a complex disease. You have over 400 loci associated with the disease. However, when you start looking at these tag SNPs, you have to consider haplotype structure of the human genome. You have to look at the linkage disequilibrium in order to count all the SNPs that are associated with the disease. And there are 12,000 SNPs. So good luck figuring out which are causal unless you do some kind of a filtering. So our filtering is based on our ThreadNet model. And when we do that, then we drop 12,000 to 52 variants that we believe has the most pronounced effect and are likely, most likely causal in type 2 diabetes. How can we validate that? RNF6 is strongly associated with type 2 diabetes. It's one of the risk factors, and upstream of this gene, there are 30 variants. So GWAS gives you 30 variants. So which one of them is really causal? We say that that's the variant. We run it through our computational pipeline and we get the variant. Without experimental validation, we just got one out of 30. We were lucky that in this case, the impact of this variant on the gene expression has been published and demonstrated that it drops the level of RNF6 expression sixfold, and it is indeed a regulatory variant. So, okay, we're reinventing the wheel, but can we detect something new, something that hasn't been published yet? That's another locus, and that's how your GWAS associations of variants look like. And there are tons of associations. If you run glucose sensitivity assays, out of those top associated variants, you get a half, saying half of them are associated, half of them are not. However, our model says that there is one, maybe two variants that are associated, that are causing dysregulation of this gene in this particular locus. That was the work of our collaborator Stitzel's lab from the Jackson's laboratory, where they took the variant that we predicted as causal, a couple of other variants that we predicted as not, and they have been able to demonstrate that in this EMSA, electromobility shift assays, this is the central variant is the variant that causes the difference. You start looking at luciferase reporter assays, exactly that's the variant that changes the level of gene expression, not the other two variants. So there is some sense that the predictions make sense from the functional viewpoint. The paper has been published last year, and now we have this toolkit where we can apply to different diseases and try to predict functional and disease causal variants. Type 2 diabetes is fine. However, we can expand that to quite a few other diseases, in reality, any. So we can take any complex disease, Mendelian disease is just way too easy, right? And try to predict disease causal variants. This is a case where we said, okay, why don't we look at neurological disorders? And we took 6 different ones. So you see schizophrenia, you see Alzheimer's, you see Parkinson's. And again, we are kind of cheating. We're saying, okay, there are loci that are strongly associated with these diseases. And within this loci, disease causal variants have been profiled. And those are red triangles. So if we don't know about that, if we go into this loci and use our predictor, would we be able to get them? And as you can see, the top scoring predictions within this loci are indeed those that fall into the red triangle, suggesting that in the vast majority of at least these small sample cases, we can accurately detect disease causal variants without knowing which one or which of them are disease causal. With that, so that's that was a long introduction. So apologies for that, because we have, as I told you, we don't care about enhancers, we care about silencers. And silencers and enhancers, they are more or less the same, right? So they are all affecting the gene expression of their target genes. Enhancers are upregulating gene expression, and silencers are downregulating gene expression. Otherwise, they're more or less the same. They're looping towards the promoters. There is this interaction, polymerase 2, etc., etc., etc. It is, however, there is a reason why people have been studying enhancers and not silencers. Silencers, it is very, very difficult to detect them from the experimental viewpoint. There are not that many histone marks and that we can use to get to them using ChIP-seq experiments. And there are not that many assays that have been developed for validation of silencers. You get a silencer, what you believe is a silencer element, it is very, very difficult to prove that it is what it is. And in order to prove my point that nobody cares about silencers, if you still doubt that, that's the number of publications addressing enhancers in one way or another in yellow, and that's the number of publications addressing silencers. And as you can see, it's really that absolutely nobody cares about silencers, despite them covering almost as much human DNA as enhancers. And maybe the reason for that is that they do absolutely nothing. Another part of junk DNA. Maybe they are. So that's a case of facial paresis, and there are two genes that are strongly associated with the disease. One of them is GATA2 transcription factor, and what has been demonstrated that in facial brachial motor neurons, there is a deactivation of a silencer. So the gene has a primarily regulatory architecture consisting of two enhancers, green and blue, and one silencer in red. When the silencer is deactivated, you get this asymmetry and of facial features, and you get a strong phenotype that's associated with disrupted binding of NRF2 F1 to this silencer element. Okay, so maybe not all silencers are junk. Is it possible to detect them? And what we've tried to do is to develop a model. I'm not going to go into the details, every dot is a cell line and this and that. I'm just going to tell you that the accuracy of silencer predictions is not as great as the accuracy of enhancer predictions. It's really difficult to get them for some reasons. We are still struggling. We can get them, but it's for some reasons it is challenging. And we think that the GC content is playing a role in this. And they're not only different in terms of the GC content, they're also full of repeats. So silencers are strongly associated with repetitive DNA in the human genome. So that makes it difficult extracting the signal. However, if you look at the genes that flank enhancers versus silencers, as you would expect, enhancers upregulate the genes, and that you see from over here, the blue region went up. That's the level of gene expression close to enhancers. When you look at the silencers, these genes are downregulated, irrespective of whether you use proximity of a high C contacts. Anything that's related to silencers is downregulated, according to the silencers that our model predicts as such. This is experimental work. Again, nobody trusts computations for good reasons until it has gone through experimental validation. And this is a silencer assay that has been developed by our colleague Laura Elnitsky's lab from the Genome Institute. And what she has been able to do is to demonstrate that 5 out of 10 silencer predictions indeed act as such. And when you look at 50% validation rate, you can kind of argue whether it's good or whether it's bad. For enhancers, it's the same, about 50%. That was the ENCODE project was showing to us, that the best you can get is about 50%, and you can look at cell type specificity and timeline of activation, this and that. So that looked fine. Going back to the diseases. So we've got silencers. We believe that we can predict silencers. Do they relate to GWAS SNPs at all? Do GWAS SNPs fall into silencers? And this is the density of GWAS SNPs in enhancers yellow and silencers blue. And all of these two densities are significantly elevated when you compare to the background. When you look at the CleanVar dataset, which is a curated subset effectively of GWAS, you see that there are many more silencer variants, suggesting that they seem to be related to the diseases. EQTL data shows about the same. So what can we do with these associations? We can start looking into different diseases, and we can start profiling the degree of association. For, in this case, we looked at neurological diseases, we looked at dementias and their brain volume as their proxy, Parkinson's and Alzheimer's. And what we've done, we looked through a bunch of different cell lines and asked the question, is it silencers or enhancers that are stronger associated with these diseases? And what you see, for example, in the case of brain volume or Parkinson's, It's silencers. It's the disruption of silencers that has a much more pronounced effect on the disease, as opposed to Alzheimer's. In Alzheimer's, it's yellow, it's enhancer mutations, while in Parkinson's and dementias overall, it is silencers. However, the picture is kind of interesting because you can, when you look at the cell lines, you can break it down into brain development versus the immune system response. And over here, you have immune system, you have metabolic and endocrine pathways, and you have brain development in green. And in case of Parkinson's, brain development is silencers. Immune system response is enhancer mutations. So it looks like there is a complex interplay between silencer and enhancer when you start looking at disease causal variants. We can go ahead and move from cell types into different genes. And in the case of Parkinson's, we looked at the genes that our model predicts as the most associated with the disease. And if what you see here, you see a bunch of genes, and these are gene names. And if you see the association with blue dots, it's their association with silencers. So silencers within the loci of these genes are dysregulated. Red ones are enhancers, and in between, it's a mix. And two hallmark Parkinson genes, this TRIM40 and TRIM31, they are full of silencers in their loci. And those are silencers that cause GWAS mutations, suggesting that those have been driving the dysregulation of these genes. These genes are upregulated in Parkinson's. And this is one of the examples when you talk about the mechanisms of the disease. You have a locus of a gene of CRHR1 that's associated with Parkinson's, and two SNPs have been previously recorded as risk alleles of the disease. And those are red bars. When you look at those and overlap with silencing activity in blue or enhancer activity in yellow, you see that across multiple kinds of cell types, it is silencing activity that's overlapping these variants. And what we've been able to demonstrate that these variants, they disrupt binding of repressors. And SNP-Selix data shows that when you disrupt binding of a repressor transcription factor to one of the silencers, sometimes it is also accompanied by creation of an activator. So you have some kind of a reprogramming of silencer into an enhancer. And we can go into other diseases, schizophrenia, bipolar disorder, and we can quantify the contribution of silencers versus enhancers. Schizophrenia, it's mainly enhancers. Bipolar disorder, it's mainly silencers. And again, the picture can be broken down into the contribution of the immune system versus brain development. That's type 1 diabetes and arthritis, and we can kind of repeat and identify key contributors for different diseases. And in this case, I'm not going to repeat myself. I'm just going to tell you that silencers are playing an important role in type 1 diabetes. But when you look down and try to figure out what are the mechanisms, what you end up identifying is this locus. I don't know where it is in particular, but it's an insulin IGF-2 locus, where a mutation that we predict as a silencer mutation, what it does, it damages that silencer and reactivates these genes. And what's known about type 1 diabetes is that IGF-2 overexpression is associated with immune system onslaught that leads to type 1 diabetes. So somehow, by applying this model, you can narrow it down to the mechanisms of diseases from just the identification of disease causal variants. Cancers, I know that quite a few labs here work on cancers. And if we ask about the role of enhancers in yellow versus silencers, we can see that across multiple cancers, we can identify a comparable role of silencer mutations to enhancer mutations to different kinds of cancers And for myeloid cancers, it's male enhancer mutations. For breast cancer, it's male enhancer mutations. And we can look down into different diseases a little bit more granularly, but I'm almost done. Let me tell you about something else that's based on silencers, but not necessarily silencers. You've heard about super enhancers, but have you heard about super silencers? Apparently, there are super enhancers in our genome. And as many as there are super enhancers, there are super silencers. So you have genes that are blocked strongly, not by a single silencer, but a huge cluster of multiple silencers working together. And for some reasons, 1,000 of genes is blocked by super silencers. And you could ask why? Why do you need such kind of a block? When you look at gene expression, and this is the background, this is the level of gene expression close to silencers, enhancers, super enhancers, and super silencers. As you can imagine, genes close to super enhancers are super activated. They express really at high levels. Super silencer genes, they're really well super downregulated. So it's a reverse effect. When you look at the regulatory activity using ATAC-STAR-seq data, you observe that the effect again is opposite of silencers, super silencers, enhancers, and super enhancers. The same lab that we worked with prior, they took our prediction of super silencers and demonstrated that they do consist of individual silencer elements that work as such in silencer reporter assays. And then we're going to go into the details of that. However, what I would like to tell you is that when we start looking at mutations that fall into silencers versus super silencers, and we look at GWAS variants, we see significantly more silencer and super silencer GWAS variants when we look at B-cell cancers. So it's not all GWAS. GWAS densities are comparable. But when you go into a set of B-cell cancers, you get just some mutations in enhancers and super enhancers, and tons of them in silencers and super silencers. Recurrence, the recurrent mutation, many of them are. And what we can do, we can predict these mutations and look at the genes that are located next to these super silencers. And those genes, if they have this red asterisks next to them, they are known to play a key role in B-cell cancers, including BCL6 and BAC2. So let's go back quickly to the question of why might you need a super silencer? What you look at here is the progression of B-cell maturation, starting with embryonic stem cells, going through mesoderms and HPCs. And what you're looking at the levels of gene expression. Reddish colors are enhancers, bluish are silencers. Let me summarize that for you. What happens is that genes that are associated with super enhancers, that red line, they're kind of not expressed during early development. These are the genes that are going to be highly expressed due to the activity of super enhancers and B cells, and they will be highly expressed later on. So it's kind of a progressive increase. However, when you look at the super silencers and see the genes that in mature B cells are downregulated, what are these genes? These genes were not at zero. These are the genes that were super enhanced. Yes, they were next to super enhancers in embryonic stem cells. And in order to super suppress them or reverse the activity of super enhancer, you need, or it appears like you need a super silencer to reverse that role. So what happens when that super silencer is damaged? And our data seems to suggest that damage of a super silencer leads to reactivation of a super enhancer and cancer progression. because you somehow reverse the fate of the cells back into their developmental state. Is it only due to the accumulation of mutations? Our data seems to suggest that no. We also see a lot of translocations. For example, BCL6, 35% of DLBCL cases are associated with translocations. And what do these translocations do? These translocations we can demonstrate in case of BCL6. They take that super silencer, chop it into pieces, and bring it into a super enhancer locus. And that's going to reactivate the expression of BCL6, and that seems to be associated with DLBCL. Is it recurrent? Yes. If you look at the patients, we see recurrence of these translocations and cases where super enhancers are changed to super silencers. We see much stronger effects across the patients with DLBCL. And we asked the question of how does it happen? So what we know about DLBCL is that there is a transcription factor and cofactor called BERD4 that plays a role in activation of super enhancers, and especially super enhancers in DLBCL. We know that not all of these super enhancers that are active in B-cell cancers, in particular in DLBCL, have the same nature. Some of them appeared from nowhere. Some of them originated from enhancers. And some of them represent these transitions of super silencers to super enhancers. You have one of them which is being deactivated either due to mutations, translocations, or epigenetics. And we ask the question, if we see that case of super silencers being translated into the super enhancers, does it have anything to do with this disease? And we observe the strongest association of these elements with the activity of BRD4. And what we see is if a treatment of JQ1 is applied, which is known to suppress BRD4 expression that is now reactivated due to this negative effect on the super silencer in the locus of BCL6, we can demonstrate that this treatment is strongly associated with reactivation of suppressive effects in the locus of BCL6, and you can start seeing these effects shortly in 2 hours after the JQ1 treatment. And the last slide that I have is for computational people in the room. And it has nothing really to do with silencers, but I get asked often, why do you use rather simple deep learning models? Okay? Now there are simple and there are not simple deep learning models. If you use ChatGPT, you know that the whole LLM field, large language models, is based on transformers. And this is a new architecture that supposedly all of us should be using. And it looks like it works so well for so many different tasks that relates to human speech and human languages. But when we compare our model, that's primarily a CNN model, to another CNN model, to transformer models that are in blue and hybrid models that are in orange, we can demonstrate that the performance of CNN models is much, much better in application to genomic, the second language of the genomes for some reasons. So just a take-home message that LLMs work great for the human language, not that great for the genomic language. And with that, I would like to thank Ji Huan, a staff scientist from my lab, who's done the bulk of the work that I presented today as well as our collaborators from the Genome Institute, Elnitsky and Collins groups, Len Pinocchio's group from the Berkeley lab, Evgeny Kvon's group from UCR1, and Stitzel's group from the Jackson's lab. And thank you so much for your attention. Thank you so much for coming to this seminar, and I would be more than happy to answer questions if there are any. Thank you.
Thank you very much for your super interesting talk.
Thank you.
So please question. Yeah, we'll start then with the ask all questions. So in terms of, I am not very familiar with variants for non-coding regions. I'm more familiar with, like I said, disease-associated variants for coding regions. But it looks like, of course, non-coding region occupies the vast majority of genome. 20% of genome are occupied by enhancers. Are there any mechanisms which sort of prevent the deleterious effects of these variants in terms of redundancy of enhancers, or maybe some mutation rate composition of these enhancers, so that, because the probability that some of these enhancers will be mutated and the whole thing will be dysregulated is pretty large. And I wonder in cancer, well, because there are non-coding variants causing variants in cancer, but mostly I think we know about coding variants. But it seems like for cancer, this is pretty good, I would say, straightforward mechanism to dysregulate pathways to actually introduce mutations in non-coding regions.
And that's a very good point. On average, a human gene has 5 enhancers. So obviously, some genes have hundreds, some genes have only 1, but on average 5. And if you are absolutely right that about 20% of the human DNA is regulatory in nature, but regulatory is the term that everybody uses to say enhancers. We find almost as many silencers. So you have a gene, and on average, you have 5 silencers and 5 enhancers. And no, the impact of all of them is quasi-redundant. It's additive. What we know, it's additive, but it could be that one enhancer is shadowed by the other ones. And a mutation can destroy one of them, and you would have 0 or non-quantifiable impact on the level of gene expression. And that indeed happens left and right. So often you do have to have multiple mutations that affect multiple enhancers. However, it's not the case for all the genes. So for some of the genes, there are, you can call them driver enhancers, you can call them key enhancers that will demonstrate contact the promoter the most, and other regulatory elements channel their activity through that key regulatory element in the locus. So if the mutation falls into that one, that is going to have the most pronounced impact on the level of gene expression. However, again, it depends on the locus, it depends on how many enhancers do you have and what is the role of every single one. And in regards to cancer, what we observe is that overall, we think that the role of silencers is greater, or at least according to all our computations, is greater in cancer progression than the role of enhancers. So what our data seems to demonstrate, or at least across multiple cancers that we looked at, not all of them obviously, is that you have quite a few genes that are suppressed. And if these mutations start reactivating the genes, if these are developmental genes, that seems to be associated with cancer progression, and especially those mutations affect super silencers.
Question from students? No?
By name. Asking by name.
Everybody's tired.
So I go back a long way in this field, and I remember when enhancers were first discovered. But after that, there was that idea that there were insulators associated with enhancers, and the insulators were simply areas of the genome that were keeping heterochromatin from entering into
Mhm,
enhancer
mhm,
areas.
mhm, mhm.
And so serve many of the same functions. And so I guess my question is, are silencers the same as insulators? And is there a functional differentiation between those two? Are they separate elements? Or is it, in fact, is it a moot point that they're functioning as elements that are regulatory in a negative way, either whether it's de-repression or active repression, doesn't really matter.
Absolutely. And that was the first question that we asked.
Mhm.
It's kind of, is it just heterochromatin, right?
Right, exactly.
Maybe it just does nothing. It just prevents enhancers from acting.
Right.
And it's not contacting promoters. So
Mhm.
we started computing high-C contacts. And
Right.
we see that they loop towards the promoter regions.
Mhm.
Then the next question that we asked, we said, okay, if these are active elements, as opposed to the rest of H3K27 trimethylation regions in the genome,
Mhm.
what should be different is that they should be bound by transcription factors.
Right.
So we started counting the number of transcription factor binding effects using ChIP-seq data. And we observed that they are bound 4 times more often than what you might be calling insulators.
Mhm.
So our data seems to demonstrate they're active. They loop towards the transcription start sites to the promoters of the regions and have an active role in suppressing
Mhm.
Call2 activity.
Yeah.
So I think yes, it is very important.
Yeah, but I think you've answered the question.
Okay.
So, another question. So I wonder in terms of transcription factor binding sites, how sort of densely they are packed in these enhancers and silencers, because you showed this experiment where 70% of SNPs in enhancers, they actually led to the enhanced binding with transcription factors. Was it?
So 70% of them had an upregulating effect on enhancer activity. So they strengthen the enhancer activity. So let me answer your question. So what we see is that you have an enhancer. The current estimate that enhancer is 400 base pairs long, right? I'm giving you an answer that's imprecise on so many levels, but an estimate is 400 base pairs long. There are 5 binding sites within it. Again, it's an average of the averages. Each binding site is about 8 nucleotides. So 40 nucleotides out of these 400 is covered by regions that are bound by transcription factors. And then those are used by enhancers. What we see is that I spoke briefly about super enhancer to super silencer transitions. This is based on enhancer to silencer transitions. When we look at cellular development, what we observe is that half of the enhancers become silencers, and half of the silencers become enhancers. And by that, I mean exactly the same region of DNA, exactly same 400 base pairs are used as enhancer, let's say in embryonic stem cells. And if it was an enhancer, it's a 50% probability that exactly the same region of DNA is going to be a silencer. Mechanistically, when we map binding sites, is it the same binding sites? When we ask that question, the answer is absolutely not. So there are 2 codes within the enhancer sequence, a set of activator binding sites and a set of repressor binding sites. So depending on the cellular context, I think, depending on the availability of transcription factor proteins, they can act either as upregulators or downregulators of gene expression. So the genome is complex in terms of not just the composition of regulatory elements, but the same kind of regulatory elements being able to work one way and another.
Can I follow up on that? So in
Sure,
that
please.
context, is there a common set of transcription factors to the 2 states, and then some transcription factors change? Or is it a complete rearrangement of the factors that are binding?
it's closer to a complete rearrangement.
complete rearrangement.
Uh-huh.
There's no commonality, there's no, you know,
The,
overlapping links.
in biology, everything is kind of,
Yeah.
there is some commonality, but primarily it's a new set of transcription factors that acts on the same genomic regions. We see binding of repressors, those transcription factors that we can characterize as repressors,
Mhm.
acting on the same regions while they were bound by activators prior.
Now, do you see that at the level of SNPs as well? You know, SNPs that are acting on one element within that subset that have a different effect, like a repressive effect or an activator effect, and basically parsing those sites?
So in one case, we have been able to go down in a little bit more details, and these are repressor binding sites,
Mhm.
and this is an activator.
Right.
So what happens with that mutation, it removes the binding site of one of these two repressors and creates a binding site of an activator.
Right.
So I don't know how common it is. We haven't been able to characterize it. Honestly, we don't have enough data to characterize that. I would guess that in many cases, it's just a disruption of activators or disruption
Right.
of repressor binding
Yeah.
sites. But in some cases, it is a switch. Within that enhancer, it is a switch from one side to another side. Another type of a site.
One more
Okay.
question?
Just to clarify with the large B-cell lymphoma, basically what's happening is you have these mutations in the super suppressors, which cause it to go back to stem cell-like state. I was just wondering if you've ever worked with any like glial cell-based tumors as well, because this is going back to another lecture that we had probably about a year ago. There's some mutations that occur in H3K27 that actually cause kind of a similar effect, more or less. The patients, the cells, the glial cells will regress and kind of take on more of a stem cell-like state from which they can't differentiate. So I'm just wondering if you've seen anything similar inside of those types of tumors.
So our exposure to cancers has been limited so far. So we've done a little bit with DLBCL, this diffuse large B-cell lymphomas, and now we're working on ovarian cancers. And not because we like them, but because our collaborators work on ovarian cancer. But in the case of ovarian cancer, we observe a very, very similar effect where we see a large scale reprogramming of super silencers or effects where super silencers are either mutated or have been translocated, and that's associated with patients with ovarian cancer.
Yeah. Oh, yeah, yeah.
So, when we think about cancer development, we think about, sort of global hypomethylation and then hypermethylation at tumor suppressors, for the most part. In terms of that mechanism, in terms of that specificity, do these silencers, do you think, play a direct role in that hypermethylation of tumor suppressor genes? Because it seems obvious that, if you're selecting, key genes in the genome that are going to give you a growth advantage, but there has to be some sort of mechanism for basically bringing about the active suppression through this sort of process and then eventually hypermethylation.
Absolutely, yes. So I'm not rejecting hypermethylation in cancers. I'm not saying that everything is due to super silencers. It's not the most, but I think there are complementary mechanisms, and I think in order to, again, I should probably not even speak in this audience about cancers, given how little I know about that. But my feeling from what we have done is that super silencers play a non-negligible role in cancer progression, and that if we ignore them, we might be missing on a component of that.
My point is, could super silencers be the mechanism for suppressing tumor suppressor genes that ultimately leads then to their methylation as a stepwise progression?
Yeah. Right. So I would guess yes, but that's going to be just a guess.
Yeah. Who's on your next NIH grant?
I don't get grants.
You're a lucky man.
I know that. I spent over a decade writing grant proposals. Mhm.
I'll tell you how long I've spent writing paper proposals. Thank you.
Thank you so much.
So if there are no questions, maybe I encourage students to, after the talk, if you are shy, come over and ask questions individually. This is a great opportunity for Ivan, while Ivan is here, it's a great opportunity for students to interact. So thank you so much, Ivan, again.